Science has explained a great deal about how plants grow. We know the chemistry of photosynthesis down to the molecular level. We know how root hairs absorb water and minerals through osmotic gradients. We know the hormonal signals that trigger germination, flowering, and fruit set. We have mapped the genetic sequences of hundreds of plant species and identified the specific genes responsible for leaf color, fruit flavor, and disease resistance. And yet, there are things that happen in gardens that science has not fully explained. Observations that have been repeated by gardeners across centuries and across continents that do not fit neatly into what we currently understand about plant biology. Phenomena that are real, documented, and reproducible, that experienced gardeners accept as facts of gardening life, and that researchers have studied without arriving at consensus explanations. This is not mysticism. It is not an argument for abandoning the scientific understanding of plants that has made modern horticulture possible. It is simply an honest acknowledgement that our understanding is incomplete, and that some of what gardeners have observed for generations is more interesting, more complex, and more resistant to simple explanation than the tidy narratives of popular plant science suggest. Here are 7 of those discoveries. Before the 7 discoveries themselves, one framing observation that gives them context. The history of plant science is full of examples where gardener observations preceded scientific explanation by decades or centuries. The observation that specific plants grow better together predated the discovery of allelopathy and mycorrhizal communication by millennia. The observation that wood ash improves soil and plant performance predated the understanding of pH chemistry by centuries. The observation that composted organic material produces better garden results than the same organic material used fresh predated the understanding of soil microbiology by generations. In each case, empirical garden observation was correct before science understood why. The 7 phenomena in this video are in that tradition. They have been observed reliably. The explanations are incomplete. That does not make the observations wrong. Here are 7 of those discoveries. Discovery 1. Plants respond measurably to sound. This is one of the most studied and most contested observations in plant science. The basic phenomenon is well established. Plants exposed to sound waves of certain frequencies show measurable differences in growth rate, germination speed, root development, and stress hormone production compared to plants grown in silence. The research extends back to the 1960s and has been replicated in peer-reviewed studies across multiple countries, plant species, and experimental conditions. What science has not explained is why. Plants have no auditory organs. They have no nervous system. They have no known mechanism for detecting and responding to sound pressure waves in the way that this research suggests they do. One proposed mechanism involves the mechanical stimulation of tiny ion channels in plant cell membranes by sound vibrations, which could trigger biological responses similar to those triggered by physical touch. This mechanism is biologically plausible, but has not been definitively demonstrated. Another proposed mechanism involves the effect of sound vibrations on water movement in plant tissues. Neither explanation has reached scientific consensus. What gardeners have observed across centuries, without scientific frameworks to explain it, is that plants near music, near conversation, near the sounds of human daily life, often appear to perform differently from plants grown in silence or in noise. Some of the oldest accounts of talking to plants as a garden practice come from cultures that had no framework for explaining why it might work, and no interest in anything other than the empirical observation that it seemed to. The observation predates the research by centuries. The research has confirmed the phenomenon without explaining it. One specific study that illustrates the depth of this unexplained observation was conducted at the University of Missouri, where researchers found that plants respond differently to the sound of caterpillar chewing, specifically producing elevated levels of defensive chemical compounds in response to the sound vibrations produced by feeding insects, even when no actual insect was present, and only. A recording of the sound was being played through the plant's stem. The plant appeared to be responding to the sound of a specific threat, rather than to any chemical signal from the threat itself. The researchers could demonstrate that the response was real and measurable. They could not fully explain the mechanism by which a plant without a nervous system was distinguishing between the sound of chewing and other sound vibrations and generating a specific defensive response to one and not the other. Discovery 2. The moon appears to influence plant growth and garden outcomes. Lunar planting calendars have existed in virtually every agricultural culture on Earth for thousands of years. The basic premise is that the phase of the moon at the time of planting, harvesting, or other garden activities affects the outcome of those activities. Root vegetables planted in specific lunar phases germinate and develop differently from the same vegetables planted at other phases. Harvested crops stored at specific lunar phases keep longer than those harvested at other phases. The proposed mechanism involves the moon's gravitational influence on water within the soil and within plant tissues, similar to the tidal effect on large bodies of water. If the moon can move oceans, the reasoning goes it can move the smaller quantities of water in soil and in plants. The gravitational effect on volumes of water as small as those in a garden bed is calculable and is genuinely small, but it is not zero. Whether this small effect is sufficient to produce the differences that lunar planting proponents report is a question that physics can calculate approximately, but that the research literature has not settled. What makes the lunar planting observation particularly interesting is its global consistency. Cultures with no historical contact with each other developed independently similar observations about the relationship between lunar phase and plant behavior. The parallel development of the same observation across cultures that could not have shared information about it suggests either a genuine phenomenon or a globally consistent cognitive bias. Both possibilities are interesting. Neither fully explains the observation. The specific lunar planting detail that generates the most consistent anecdotal support among experienced gardeners is the recommendation to harvest crops for storage during the waning moon rather than the waxing moon. Vegetables harvested during the waning phase are reported to store longer and maintain their quality better than equivalent vegetables harvested during the waxing phase from the same bed on alternate days. This is the kind of specific, testable, repeatable claim that should be easy to evaluate scientifically, and yet controlled studies on the subject have produced inconsistent results that leave the phenomenon neither confirmed nor definitively disproved. The inconsistency of the research results may itself be informative. If the effect is real but small, and if it interacts with other variables in ways that controlled studies do not account for, the kind of inconsistency observed in the research literature is exactly what you would expect. Discovery 3. Some plants appear to communicate distress to neighboring plants before the physical symptoms of that distress are visible. This is one of the most active and most fascinating areas of current plant science, and it is one where the research is genuinely surprising. When certain plants are attacked by insects or pathogens, they release volatile organic compounds into the surrounding air. Neighboring plants that detect these compounds have been shown to respond by upregulating their own defense chemistry, producing pest-deterring compounds before any pest has reached them, as if they received a warning. The mechanism for this airborne chemical communication has been identified in broad terms. Specific volatile compounds, specific receptor systems in neighboring plants, specific chemical defense responses. What has not been explained is the sophistication of the communication. The volatile compounds released by a stressed plant are not uniform. They vary based on the specific type of stress the plant is experiencing, and the responses of neighboring plants vary in ways that are specific to the type of stress signaled. The chemical conversation between plants appears to be more specific and more information-rich than a simple alarm signal, and the biological mechanisms for encoding and decoding this level of chemical specificity are not fully understood. What gardeners have observed in the form of the old companion planting knowledge that certain plants do better near each other and worse near others may be partly an observation of this chemical communication system in action. Plants that grow better near each other may be communicating and responding in ways that benefit both. Plants that consistently underperform near each other may be in chemical conversations that are less mutually beneficial. The companion planting knowledge that predates the research by centuries may be recording the outcomes of plant communication that we are only now beginning to understand mechanistically. One of the most striking examples of plant communication documented in recent research involves tomato plants and spider mites. When spider mites begin feeding on a tomato plant, the plant releases specific volatile compounds. Neighboring tomato plants that detect these compounds respond by upregulating jasmonic acid production, which triggers the synthesis of defensive proteins that inhibit the development of spider mite eggs and larvae. The defense response happens before any mite has reached the neighboring plant. The neighboring plant is preparing for an attack that has not yet arrived, based on chemical information received from a plant that is under attack. The speed, specificity, and effectiveness of this response exceed what a simple alarm signal would produce and suggest a level of information transfer between plants that current models of plant biochemistry do not fully account for. Discovery 4. Plants grown from seeds saved by the gardener often outperform the parent plant in that specific garden. This is not simply a matter of acclimatization, though acclimatization plays a role. Gardeners who have saved seeds from their best-performing plants over multiple consecutive seasons in the same garden location consistently report that by the third or fourth generation, the garden-saved variety outperforms any purchased variety of the same plant in that specific location, even when the purchased variety is from premium sources and the saved seeds are from ordinary plants. The mechanism should be straightforward natural selection. Plants that perform better in a specific environment leave more seeds, which inherit the characteristics that made the parent successful in that environment. Over multiple generations of selection pressure in the specific garden microclimate, the seed-saved population should become better adapted to that microclimate than any commercially produced variety bred under different conditions. This is the theoretical explanation, and it is plausible. What makes it surprising is the speed. Natural selection typically requires many more generations to produce measurable phenotypic differences than the 3 or 4 generations that experienced seed savers report as sufficient to observe meaningful local adaptation. The speed of the adaptation in garden settings exceeds what simple genetic selection would predict, which suggests that additional mechanisms, possibly epigenetic changes that are inherited across generations, may be involved. Epigenetic plant inheritance, the passing of environmentally induced gene expression changes from parent to offspring, has been documented in plant research but is not well understood as a mechanism for rapid local adaptation. The practical implication of this observation for seed savers is significant and worth making explicit. If the garden-specific adaptation that experienced seed savers report is real, and if epigenetic mechanisms are partly responsible for its speed, then the act of saving seeds from the most successful plants in your garden each season is not simply preserving genetic material. It is actively shaping the gene expression patterns of the next generation in response to the specific conditions of your specific growing environment. The seed that performs best in your garden in year 4 of consistent saving is not just genetically similar to the parent that produced it. It may carry epigenetic markers that specifically prepare it for the conditions you garden in a way that has no equivalent in commercially produced seed that has not been exposed to your specific environment. Discovery 5. Grafted plants sometimes take on characteristics of the rootstock that have no genetic explanation. Grafting is one of the oldest and most widely practiced techniques in horticulture, used for fruit trees, roses, tomatoes, and many other species. The conventional understanding of grafting is clear. The rootstock provides the root system and determines drought tolerance, disease resistance, and soil adaptation, while the scion provides the above-ground growth and determines the fruit, flower, and vegetative characteristics. The genetic material of the rootstock and the scion remain separate. What has been observed repeatedly in grafted plants is that this separation is not as complete as the genetic model suggests. Scions grafted onto different rootstocks sometimes show differences in fruit flavor, fruit color, and even leaf morphology that cannot be explained by the genetics of the scion alone. These are characteristics that should belong to the scion's genetics, not the rootstocks, but they appear to shift when the rootstock changes. Research into this phenomenon has identified small RNA molecules that can move between rootstock and scion across the graft union and potentially influence gene expression in both tissues. The mechanism is real, but the extent of its influence on observable characteristics like fruit flavor and color is not fully established. What experienced orchardists have known for generations, that the rootstock does more than provide roots, is being slowly confirmed by molecular plant science that is still working out what exactly is being communicated across the graft union and how. Discovery 6. The same variety of plant grown from seed can produce dramatically different results in adjacent garden beds with apparently identical conditions. Every experienced gardener has encountered this. You plant 6 tomatoes of the same variety from the same seed packet in the same row. 5 of them grow to the same height and produce a similar yield. The 6th grows significantly taller, produces significantly more fruit, and shows notably more disease resistance than the other 5. The seed was from the same source. The conditions were the same. The care was the same. The standard explanation involves genetic variation within open-pollinated varieties, where individual plants within the same named variety carry different versions of genes that produce different phenotypic outcomes. This is real and accounts for some of the observed variation. but the degree of variation that experienced gardeners consistently observe within the same open pollinated variety, and the consistency with which specific spots in a garden produce better results than adjacent spots with apparently identical conditions, suggests that something beyond simple genetic variation is at work. Microbiome variation in garden soil, which can differ dramatically between spots that are physically close and visually identical, may be part of the explanation. Specific combinations of soil organisms in a particular spot may interact with specific genetic variants in a particular plant to produce outcomes that neither the soil nor the plant genetics would predict alone. This remains an active area of speculation rather than established science. Discovery 7. Plants appear to anticipate seasonal changes before the physical triggers of those changes are measurable. Trees begin preparing for leaf drop weeks before the temperature drops that trigger abscission. Bulbs begin their underground spring growth during the coldest weeks of winter, before soil temperatures have risen to any level that should be triggering growth. Annual plants time their seed production to the exact period that maximizes germination success in the following spring, which requires an accurate internal prediction of when the current season will end and what conditions the next one will bring. The mechanism for this apparent anticipation is understood in broad terms. Photoperiod, the length of daylight, provides the primary timing signal, and plants have internal biochemical clocks that integrate photoperiod information with temperature history to make timing decisions. This much is established plant biology. What is less well understood is the precision of these timing systems in natural conditions. Research on plant phenological timing has repeatedly found that plants respond to seasonal change with a sophistication that exceeds what the known signaling mechanisms would predict, as if they are integrating information from multiple signals simultaneously, in ways that our current models of plant biochemistry do not fully account for. What gardeners have observed as a kind of plant wisdom, the sense that experienced gardeners describe of plants knowing what to do and when to do it without being told, reflects this genuine biological sophistication. Plants are not intelligent in any recognizable sense, but they are, in ways that we are still working to understand fully, remarkably good at predicting what comes next and preparing for it. The precision of plant phenological timing is most visible in its failures, which occur when the timing systems produce the wrong prediction. A warm December that fools bulbs into early emergence followed by a hard January freeze kills the bulbs that responded to the false spring signal. A late spring frost that arrives after the phenological system has already committed trees to bud break causes the frost damage that is visible in a late spring orchard. These failures reveal how the system works by showing what happens when the prediction is wrong. And they also reveal that the prediction is usually right, because plants have been making these seasonal predictions successfully for millions of years in environments where a wrong prediction was fatal, which is exactly the kind of selection pressure that produces the apparently sophisticated anticipatory behavior that gardeners observe and that science is still working to explain. These 7 phenomena are reminders that the garden is not a simple system, and that the gardener is not its only intelligence. The plants have been at this longer than we have. The things they do that we cannot fully explain are not failures of our observation. They are invitations to pay closer attention. Drop a comment telling me which of these 7 discoveries you have personally experienced in your own garden and what you observed. I read every comment. Give this a thumbs up if it made you think about your garden differently, and subscribe for plant science and gardening content that asks the deeper questions every single week.